Hello, everyone, and welcome back to Better Biopharma. I'm your host, Tyler Menichiello, and I'm so pleased to be joined today by Dr. Michael Roberts, co-founder, president, and CEO at Adaptin Bio, as well as Dr. Mustafa Khasraw, medical oncologist and physician scientist at Duke University. Dr. Roberts, Dr. Khasraw, thank you so much for joining me today.
Pleasure to be with you, Tyler. Thank you.
Thanks for having us.
Of course. For those of you out there who don't know, Adaptin Bio is finding a way to overcome the challenge of delivering drugs to the brain to treat CNS indications. And their lead asset is APTN 101, which is a bispecific T cell engager designed to help shuttle or hitchhike T cells across the blood brain barrier to treat CNS indications. So I really just wanted to open up this conversation. Dr. Roberts, we could start with you. If you could tell our audience a bit about Adaptin and your brain's bispecific T cell engager, the BRiTE platform.
Yeah, I'd be happy to, Tyler. Adaptin Bio is a 4-year-old company. The co-founder, Simon Petter, and I set out to, what you mentioned, overcome some of the limitations in medicine, which happens to be delivering drugs into the brain. There's this thing called the blood-brain barrier, which prevents medicines from entering the brain because it's there to protect the brain. But unfortunately, it prevents drugs from getting in. So we investigated various technologies across the country. We identified the group at Duke who was using what we call the BRiTE technology to deliver a T-cell engager into the brain. And we liked this because they were developing it for a highly aggressive form of brain cancer called glioblastoma. And both Simon and I had friends and family that have passed away of glioblastoma. So now we have an interest in actually seeing a treatment being developed for this disease. So we licensed the technology, brought it into the company, and we've been developing it ever since. So the technology is called BRiTE. It's spelled BRiTE, which is for brain bispecific T-cell engager. It's a 2-component system, where the first component is a bispecific antibody, and this bispecific antibody targets a tumor-associated antigen on the surface of tumors. The other part, the other half of the bispecific, is a anti-CD3, so it binds to the CD3 receptor complex of T cells. And when this bispecific antibody links the T cell to a tumor cell, it instructs the T cell to kill the tumor cell. So it's very highly specific. And so the initial drug we're developing is APTN 101, which targets an antigen called EGFR-V3, which is very specific to tumor cells. It's not expressed on other cells in the body. So it's highly specific. And therefore, now we can target, the brain cancer into the brain. Now, the problem with T-cell engagers and brain cancer is that this is a 50,000 Dalton protein. It's fairly large, so it doesn't enter the brain efficiently. So we need a delivery vehicle to get it there. So we know that T-cells in generally don't cross the blood-brain barrier unless they're called to do so, but they do get there. And so what we do is we take a T-cell from an individual, we manipulate it, activate it, so that it expresses certain receptors on its surface, so that it now has the ability to cross the blood-brain barrier. And when you combine the T-cell, manipulated T-cell, with the T-cell engager, they combine and it transports the T-cell engager into the brain, okay? Now, we typically use the T-cell as a delivery vehicle. But the good thing about the way we're doing it here is that in particularly glioblastoma, which is highly immunosuppressive, a T-cell engager is only as good as the T-cells that they activate. And in glioblastoma, T-cells are generally anergic, lack function, things like that. So what we do with our delivery vehicle, we're now delivering a highly functional T-cell to the tumor microenvironment, where now the T-cell engager can highly activate these T-cells against the tumor cells. And so we think that the combination of the engager, the delivery vehicle, and combining the two will make a huge impact on the treatment of glioblastoma in the future.
Yeah. Thank you, Dr. Roberts. And you mentioned before about folks at Duke, and so Dr. Khasraw, this is probably a perfect opportunity to ask what is the relationship between Adaptin and Duke, and if you wouldn't mind speaking to your work in the field and how you think BRiTE compares to other delivery solutions to bringing drugs into the CNS.
Yeah, thank you. Duke has one of the largest brain tumor centers in the country. We see about close to 1,000 patients with newly diagnosed brain tumors a year, which is much larger than anywhere else in the United States. And we also have a very sophisticated, comprehensive research program ranging from cancer epidemiology to immunology to target discovery and clinical trials. This technology that we are talking about was initially developed at a lab at Duke, John Sampson's lab, and then it was further developed by several investigators. And most recently, myself, I have been working in the field of investigating T cells in the brain and how to make them more active. Like Michael mentioned, the immune cells that specifically kill cancer cells or responsible for killing cancer cells are T cells. But in the brain, we have evolved to protect our immune system from attacking our brain. So if you have overly enthusiastic T cells, you get conditions like multiple sclerosis. We don't want that. And that's how the brain is protecting itself. Unfortunately, in the case of a brain tumor like glioblastoma, we want the T cell to change their behavior and become active again to attack the cancer cells. And that has not been very straightforward because this mechanism to protect the brain has evolved over millions of years, and we want to change it back to attack the cancer cells very rapidly when the patient develops tumor. So in this case, our research has been focused on how to re-energize the so-called exhausted T cells, and not just that, but also make them traffic and preferentially go to the tumor area in the brain. And this is the basic research that we have been focusing on. But I think the partnership here is also unique because we take science from the bench all the way to the patient. So we are taking a technology, a scientific concept that was developed in a basic science lab, and now we are at the point of taking it to patients in a clinical trial. We have been working on all the like hurdles to get this to the patient, but we're also seeing that as an opportunity to serve an unmet medical need. Like Michael said, glioblastoma is one of the most um, devastating cancers, not just because the cancer itself is aggressive, but because it grows in the brain, it affects the identity of the person. So people can't think, impairs their neurologic function, they lose their independence. So it creates a significant burden, not just for the patient, but for the family, the carers, the society. So I think this will be very impactful.
Yeah. Thank you, Dr. Khasraw. And I'm curious, you or Dr. Roberts, whoever feels most comfortable, explaining what were the historical breakthroughs that kind of inspired this technology? I mean, how what had to happen so that we could advance to the point of BRiTE being a feasible way of, again, hitchhiking T cells across the blood-brain barrier in a therapeutic sense? And how would you compare this to other means of delivering, or, these similar platforms, these delivery platforms?
Yeah, no, maybe I'll just take a quick stab at it, and then Dr. Caswell can add. So obviously, the work was developed in the lab of John Sampson, and it's, you know, uh, unfortunately, we don't have him here to speak to directly why he started this particular technology. But the general idea of having T cell engagers was started to be developed, about a decade ago, There are now several T-cell engagers on the market for mainly blood cancers. Um, but really, for solid tumors and trafficking into the brain, there really has been no way to do this. There are other technologies like different shuttles. One, for example, is like a transferrin receptor antibody, which shuttle other proteins into the brain. This technology would not work for a T cell engager because you don't want T cells attacking the blood vessels in the brain. So we had to find a new way of doing that and understanding that historically it was thought that the brain was immunoprivileged, that T cells were not trafficking. Over the last decade, there's been now a lot of investigation into this, and Dr. Khasraw has done a lot of this as well, is how do these drugs get into or T cells get into the brain? And so when you're able to manipulate a T cell to do that, or train a T cell to cross the blood-brain barrier via, expression of certain receptors on its surface, then you can actually use it as a delivery vehicle. And so that's when we combine those two, I believe that was the kind of thought behind why Dr. Samson's lab started to develop this technology.
Thank you,
Yeah.
Dr. Robert.
Yeah, what I can add to this is that there is a lot of research going on to like tweak the function of the T cells so that it attacks the cancer without causing too much damage. But unfortunately or fortunately, biology is much more complicated. So the T cells themselves, they don't work in isolation. They also depend on the tumor microenvironment and all the chemicals that are influencing their behavior. So my group, we've been like investigating different ways to improve the function of the T cell. And I think this falls nicely with the other research we are doing. And this is all like built on the shoulders of giants. I mean, there are so many groups throughout the world who work in this field at the moment. And although we have not seen any recent approvals, uh, but at the same time, there are many publications, discoveries that are very promising in this field. That includes drugs, antibodies, small molecules, but also cell-based therapies that are attracting a lot of attention, specifically for glioblastoma.
Thank you both. And I did want to talk about because it is so novel, right? And there's not many successful people ahead of you could point to, right? In terms of this T cell engagement approach. I want to hear from both sides on the development of the product itself, APTN 101, but also the BRiTE platform. I want to hear the developmental challenges around that platform. And Dr. Khasraw, maybe we'll start with you on the clinical challenges in developing this platform. I mean, what unique kind of considerations come into play when you have these?
Yeah. So I guess we have to have an IND before we go to patients, and the IND is um, a process that requires evidence of safety and preliminary activity. So the safety part, um, I think we have sufficient evidence to know that this approach is highly likely to be safe. The efficacy will depend on how the drug is given, at what time point, what period of the patient journey, and also these BiTEs tend to have a very short half-life, so they need to be given as a continuous infusion. That creates other challenges. I was involved as an investigator of another molecule that was developed elsewhere. And initially, when there wasn't much experience, like now there's a lot of experience with the BiTEs approved for hematological malignancies, there was a lot of concern about toxicity. One of the concerns is that these drugs, if they get to the brain and they engage a strong immune response, these patients could develop cerebral edema, they could develop neurological complication, and it will be very dangerous. So in that previous old study, the patients were required to stay in hospital for the duration of the treatment. And that's immediately going to increase the cost substantially because hospital stay is very expensive. And in that particular trial, Um, like one of my fellow investigators said, the commonest side effect was grade 4 boredom because the patients were just sitting there and nothing was happening. So now we have that experience. We know that there isn't a significant safety concern. So that's a clinical challenge that we think it's out of the way because of the prior experience with the similar molecules. But those trials I'm talking about, they did not include the co-administration of activated T cells. So here we still need to be cautious because the most important thing to us is the first principle is do not do any harm to the patient. So that will be like a significant aspect of the clinical development to protect patient well-being and safety.
Thank you so much, Dr. Khasraw. Dr. Roberts, anything to add there in terms of maybe looking at it from a R&D product development standpoint? And then I do want to move into the manufacturing, but curious to hear your thoughts on challenges there.
Yeah, no, I mean, as a company, we're thinking a lot of different things, right? So the clinical program is one thing we think about, and the group at Duke, when Dr. Khasraw's group is well versed to get that done, and obviously understands the history and what we need to do there. But then, as you mentioned, things like patient access, how are we going to get this drug to patients? We have a two-component system. One is a T cell engager that we manufacture at a CMO. The other is a T cell that we manufacture at the clinical lab. And then, of course, all the analytics and talking to FDA regulators. So there's a lot of things that we have to think about as we continue the development of this program.
Yeah, that's great. Thank you. And you touched on the manufacturing bit and talk about adding levels of complication here where you're sourcing both components of the product from CDMO and clinical lab. I did want to ask you about the company's CMC strategy. Do you plan to, maintain this partnership with the clinical lab and CDMO indefinitely? Is this just to get through trials? Is there a bigger, grander picture to bring everything in-house? Can you walk me through a little bit how you think about that?
Yeah. So, as a relatively small company currently, we will be sourcing out all of these activities at CMOs or third-party labs. So that won't change in the near term. I think the only time it would potentially change is when we would potentially get a drug approval, where we'd have the resources then to either build or incorporate some of those functions within the company, if it makes economic sense to do so. But as of now, our plan is to manufacture the T cell engager at a third party CMO, and those are pretty much standard manufacturing techniques, analytical techniques that one uses for T cell engagers and proteins in general. And then the other one is a little bit more specific, because again, when you talk about manufacturing of autologous T cells, these are cells from individual patients. Not all T cells are identical, and these will be a very diverse set of T cells that we get from patients. And so it's going to take a lot of expertise and rigor to come up with a specific manufacturing process to meet specifications, et cetera. So we'll do those at specialized labs, and hopefully in the future we'll bring that internally because we think we can build a business on that type of capability.
Yeah. That part about autologous T cells, I mean, I've heard in conversations before, the variability of the starting material when we're talking autologous, it could be very wide, right? That gap. And so I'm curious to what degree that affects the manufacturability of the BRiTE platform and APTN 101. How often do you run into poor starting material in the form of patient T cells?
Right. Right. So, obviously many, and Dr. Khasraw can go into more detail on this, but many of the patients that we're going to recruit in our studies have undergone some level of chemotherapy. And therefore, they've had I guess a knockdown of their T cells, and maybe it's not as robust, and so therefore you're going to have a wide variety of T cells coming in. But the good thing with this process from IND to BLA is that early on, you basically set your specifications slightly broader. And then as you progress development, you get narrow as you gain expertise, et cetera. So unlike, say, for example, CAR T cells that use autologous cells, we're not engineering these cells. We are doing some fairly simple manipulation or activation of these T cells to get them into a state where they have the ability to cross the blood-brain barrier. And so in our hands, it's not going to be as difficult as you may have heard with, say, other T cells. So the cost, the timing, et cetera, is probably going to be dramatically reduced. And therefore, we think that based on where we've gone so far, is that even if you have a broad set of range of T cells coming in, the manufacturability of this to create something that crosses the blood-brain barrier is going to be minimal. So we haven't had any issues to date. Again, it's a relatively simple process that we're doing, but I don't think this is going to be a problem for us in the future.
Oh. And from a I was curious, you mentioned before about analytics were pretty established, it sounds like, right? Were there any kind of bespoke solutions you had to come up with? And I'm curious if it's a matter of analyzing each respective half of the platform, and then, when it comes together, it's assumed to be safe, or is there a third component to it, the actual T cell engagement aspect? Is there any part of that that's involved in your release testing or potency? And
Oh yeah. So when we obviously there's a set of testing that you do for the T cell engager. It's very simple assays of identity and specificity and structure and things like that. So and binding and so forth. So it's pretty simple. But when you test the T cells, obviously it's a whole new set of assays that you do. You do binding, you do T cell activation. You look at CD69, CD25 activation, and then when and that's just the manufacturing of the T cell itself. And then when you combine it with the T cell engager, then you're looking at other assays like cytotoxicity assays against tumor cells. You're looking at T cell activation and cytokine release. Of course, you want to understand what the cytokine release is for, CRS risk and things like that. So you'll look at IL-2, interferon gamma, TNF alpha, things like that. So you're going to do a whole new set of assays when you actually combine the T cell engager and the T cells. So there's really three different silos of analytical testing that you're going to do to understand, how these T cell and T cell engagers combine to kill tumor cells.
thank you. Thank you. Um, I wanted to touch on regulatory, um, look at this product and development process from a regulatory point of view as you pursue additional INDs, because you did just successfully get an IND to bring APTN-101 to clinic. Um, do we have any insight on when those patients will be dosed, when the first cohort will be in trials, Dr. Khasraw?
We are working through the motions to get to RB approval and update the FDA with the modifications. So, a clinical trial protocol is a living document. People ask me, when would you have the final protocol? And I always tell them never, because there's always something you want to make better, because we want the best for our patients. So we constantly get feedback and we update it. So, um, I mean, it's hard to predict the future, but hopefully we'll start the first patient early next year. I don't know. What do you think, Michael?
Yeah, I think the target is towards the end of this year. So, but again, as I said, it's you know something that we continue to push, and again, the patients will be recruited when they get recruited, but we're looking at towards the end of the year.
And to take a step back, what were the biggest challenges in getting to this point? of IND and now you're hoping to recruit and dose end of this year. What have been the biggest challenges to date?
Well, from the company perspective, I think I do want to let everyone know that Duke has done a phenomenal job at getting the program to the point where we came into this, right? Typically, academic institutions don't have the kind of background and the resources because it's more of a research orient, but they don't have the resources to do a true drug development program. But the investigators at Duke did a phenomenal job of getting to the stage where we could have the ability to file. So, it was really going through and checking all the boxes to make sure that we had everything done um, to file an IND. And of course, as Dr. Khasraw said, that changes over time what your patient population, your protocol looks like. And so you keep amending that. And so I guess the challenge is to, really get something in stone and say, this is what we're doing, rather than continuing to try to optimize something, because at some point it can just drag on and on and on. So I think it's coming with a decision that this is what it's going to look like, and then we have to execute.
You provide some examples of some of the things that you're alluding to that the Duke researchers did. Like, what's uncommon that academics generally wouldn't do on an asset like this? Or
Yeah, I mean, generally like a tox program, right? I mean, there was toxicity studies that were done. There were meetings with the FDA to understand what a clinical program would look like. There was manufacturing completed at third-party CMOs with some, in-house expertise to get that done. So, a lot of academics get it to a point of lead product, and that's kind of where it goes. But they went a step further and actually started a thorough, well thought out drug development program that any pharma company would be, excited to have at that point.
Dr. Khosrow, do you think that's a testament to Duke's philosophy on its research in biomedicine, or does that speak more to the asset itself that it was, researchers were
I think
bullish?
it's a combination. I don't think it's like one thing or the other. It just happened that we have a very collaborative environment with investigators who are very keen to move things forward. And sometimes we do things that like are not necessarily expected from us. And that comes from passion, I would say, because we care about the patients and we like, I've been in clinical practice for I don't know how many years. And it's quite disheartening to see that we have not moved the needle for a disease like glioblastoma. So that's a driving force to make us like do a little bit more or maybe a lot more than just, publishing a paper, for example, or, just say this is the lead molecule and you go and run away with it. No, we like what we do and we want to see our science get to the patient.
That's great. Um, I'm curious, looking at the platform itself, the BRiTE platform, um, assuming the best and that you pursue additional INDs in the future with this platform, I'm curious what elements of APTN-101, your lead asset, what elements of that and the regulatory framework built up around it are going to be applicable to other BRiTE constructs. What will you have to do from scratch in a sense of, what regulators will expect, and what do you think is going to transfer easily as you build your
Right.
product?
Right. So the nice thing about the technology that we have is it's modular. Okay. So for example, the T cell manipulation protocol is going to be identical for every additional T cell engager that we would develop with a different target. For example, if it's HER2 or if EGFR or if it's B7H3, all of those T cells are going to be identical. Okay. So we're not going to change that process, if you will. The CD3 part of the bispecific is going to remain identical. The only change that we are going to have in this whole process is going to be the tumor-associated antigen target. And so what we expect to do, what we have now is a first-generation technology. We are developing our second-generation technology where we will have a library of these BRiTE compounds with different targets, and we will have the ability to treat a heterogeneous tumor currently, which is not possible with precision medicine, if you will, based on cost, expense, time, etc. And so we have a technology which now can treat heterogeneous tumors like a glioblastoma. And so we think our second generation approach, which uses all the tools that our first generation has, but expands it a little bit more, is going to effectively treat and manage, not just treat, but manage potentially long-term patients with glioblastoma. So that's the hope of what our technology can bring in the future. Um, and the modular and the ability to do more things with the technology than we're currently doing.
That's great. Thank you. I hope to see nothing but success on the Adaptin side. And, for patients dealing with these conditions, it's, I'm sure, a lighthouse in the storm. Well, we're running close to the end of our time, and I don't want to take too much of your day up, but I will ask you guys the question that I ask all our guests on Better Biopharma, and that is, in your opinions, whether it's in your sliver of the industry or looking out at the whole thing as a macroscopic whole, what do you think we can do to better biopharma? How can we better biopharma?
Yeah, that's a big question. I mean, obviously, there's a lot of components to drug development, and I think every component has the ability to be better and do something different. For example, FDA regulatory agencies can speed the process up. Maybe they look at using biomarkers more efficiently. They've now put in things like the platform designation, which allow sort of these modular technologies to use already, that's data that's already been generated, which can speed up that process, particularly from a manufacturing standpoint. Clinical programs, we can be better recruiters of patients, diversity, et cetera. And, just from a research and development perspective, using AI, using, um, humanized organoids or humanized animal models better to be more efficient and more rapid at creating better medicine. So I think there's a lot of ways that we can better biopharma. And I think in all aspects, it's moving, but maybe not moving fast enough for many things. So.
Dr. Roberts, Dr. Khasraw, anything to add there? How do you think we can improve the industry, or what are you excited about, perhaps, as a physician? What are you most excited to see in the near and far
I think there is this space between academic and commercial development that's like it's a limbo zone that requires more investment from all different sources. So there are certain things that are not like that incentivized to do, like the tox studies, for example. You can't get like a high impact paper from that. So as an academic, you don't have the incentive to do that, while it's absolutely essential to move the therapy to patients. And there are many things like that example, the cell therapy. There's like a lot of work that needs to be done before you get a cell therapy to a patient. And it's not straightforward to find the resources or in-kind support or financial support to move them forward. So I think it's always, to me, it's always about the patient. And this is a good example of an area of unmet need, and to accelerate development so that effective therapies become available, more investment are needed in those areas.
Great point. Thank you, Dr. Khasraw. Um, Dr. Michael Roberts, Dr. Mustafa Khasraw, thank you so much for joining me today on Better Biopharma. We really appreciate it. To all you out there listening, thank you for tuning in. We'll see you next time. Folks, once again, that was Dr. Michael Roberts, co-founder, president, and CEO of Adaptin Bio, joined by Dr. Mustafa Khasraw, medical oncologist and physician scientist at Duke University. Thank you so much for joining us for this episode of Better Biopharma, the official podcast for Bioprocess Online. For more on biologics development and manufacturing, head over to Bioprocess Online and browse to your heart's content. I'm Tyler Menichiello, and I'll see you next time.